All right, so now I want to give a summary about the whole first module on gene expression. We started with microarrays. You will have two types of cells or files. One is the cell file, which is raw data. One is a CDF file, which has the annotation of which location that probe, what gene does it target. And then we talk about using RMA for the Affymetrix microarrays to really learn the properties of a particular probe across multiple samples in order to get a really robust readout of the gene expression level. And so RMA has a number of steps. First, it used this background subtraction to remove the background. And then it quantile normalized the data in order to compare different probe values. And I forgot to mention, yeah, so also we take a log. Remember why we take a log in here? This is to make sure that the noise of the low-level probes and high-level probes are more or less comparable, right? And then we use this medium polish. Remember that Excel example, we try to always get a medium to iteratively estimate the probe effect and the expression level. At the end, then we can estimate a good probe effect and also a good gene expression level for each sample. And in order to look at whether you are normalizing your data well or calculating your differential expression well, we use this MA plot. So the M is the difference between the two samples. The A is the average or the geometric mean of the sample. So you want to see what's the level of this difference across different level of signal level, right? So that's RMA. And then after that, we discuss using lemma for differential gene expression. And we want to probably first check whether the sample really follow normal distribution using a QQ plot. Although really, if you are using Affymetrix and you use RMA, you can just use lemma. That is pretty safe. And in order to really estimate the differential expression correctly, in addition to looking at fold change, the important thing is to estimate the variance well. Difficulty there is because you only have, say, 3 or 5 replicates, so the variance you estimate may not be very accurate. Therefore, lemma use this variance shrinkage or the empirical based approach to learn the overall variance by looking at many other genes together, right? To give you some idea to kind of make the actual variance close to the group average or group distribution. And based on the differential expression, we also need to make, this is the first time we introduce FDR. So I would say statisticians, number one contribution to biologists is to teach biologists when you have so many tests that you are doing, you have to worry about multiple hypothesis testing. So false discovery rate is a way to decide, after I do all these tests, how many differential genes do I report? How many differential pathways do I report? So FDR is an important concept to learn. And then after we calculate the differential expression, we look at clustering, right? You can cluster by the samples because they have similar gene behavior across the genes, or you can cluster the genes because they have similar behavior across different samples. And we talk about different clustering approach. For hierarchical cluster, you just kind of iteratively merge things that are close by, but there is linkage difference, right? You can do complete linkage or single linkage, which will decide how to merge different nodes. And then we talk about k-means clustering, which is kind of a partition. Every gene is only, or every condition is only going to one cluster, but not the other. This is disjoint clusters. And then we briefly talk about tight clustering, which is kind of a way of getting k-means, but eliminate some outliers and noise. In fact, later on, there are other things we talk about. PCA is really also a clustering approach. And the t-SNE is kind of at least visualizing the clustering result. You can also look at it this way. We also talk about a batch effect removal. Yeah, maybe we should also mention this CCA, canonical correlation analysis in the batch effect removal. So combat is still being used. If you read some literature, people still believe that combat is doing a pretty decent job depending on your sample in terms of removing the batch effect in even single-cell RNA-seq. But in like a bulk RNA-seq, combat seems to be working very well, especially if you know the specific batch that you want to correct. SVA is more useful when there might be confounding batch effect that you are not aware of. It's able to identify these confounding effect. But in general, you can imagine combat or batch effect as a hidden treatment that you either were aware of or you were not aware of. It's not something you normally would get just by normalizing your data using RNA. Right? Because if you can really remove that, you can also remove the actual treatment effect. So that's microarrays. And then we talk about how to annotate those genes in a differential gene expression cluster or cluster of genes. And we talk about gene ontology, and you use the David for or there are like other tools for similar analysis. And then we talk about gene set enrichment analysis, which is instead of just looking at differential genes using a specific cutoff, we just rank all the genes based on their differential expression. And then we use previously defined gene sets to see whether those gene sets are their appearance in this rank order are skewed to the left or right. Right? We can also use this to give us some insight on what the differential expression experiment is really doing. And then we talk about machine learning. Like if we try to decide whether a patient has cancer or not, or we don't know whether the patient has disease or not, or things like that. So for machine learning, I mean, we mentioned this could be a whole course. We just kind of gave you a very, very brief introduction. There are unsupervised machine learning, which basically is just using clustering, right? You can use or k-nearest neighbors or MDS or PCA. By the way, MDS and PCA and t-SNE are also kind of dimension reduction because your expression data is really high dimensional, and you are trying to just draw it in a 2-dimensional or 3-dimensional way. So once you have the clustering, you're hoping, well, this one is not trying to learn any features, it's just trying to display the data. And hopefully your two classes, your known cases, your disease versus normal are separated in different clusters. Then when you have a new sample coming in, you put it in the clustering again, then you see whether it cluster with the disease or cluster with the normal. And as we mentioned that this is sometimes semi-supervised, because if the initial clustering already cannot separate your two known classes, then sometimes you tweak the parameters to make them separate better. And that's kind of not as unsupervised as we hoped. For supervised clustering, it's basically, we know the classification. These are the cancer, these are the normal. We want to look at their difference. We want to select the features that can really separate the two classes. Then just use those selected features to predict the new sample, right? So we talk about a number of these algorithms like logistic regression. In a gene expression setting, we're trying to decide which genes are important in predicting the final classes of disease versus normal. And when you select the genes, you want to kind of penalize the number of parameters you use. And we briefly mentioned L1 and L2 regularization in order to reduce your total parameters. And then we talk about random forest, which is kind of a tree-based approach, right? Every time you try to split the tree in order to separate the cancer or the two classes better, and you keep splitting, and every time you try to find the right node to split. But random forest is you sample your data and you try many times and you use different classifier together to make the final prediction, right? And then there's support vector machine, which you tried in homework 2 by this time. It's like a one-line command. It's trying to find what's the boundary of the two classes, and only use the boundary to select features to maximize the boundary distance. So important thing for supervised machine learning is to avoid the overfitting, because you could be doing perfectly on your training data, but then when you are given the new data, maybe you are actually doing poorly. So in order to really make sure that there's no overtraining, very often you do cross-validation. Remember, we have like leave one cross-validation or threefold cross-validation, fivefold cross-validation. You just train your classifier based on majority of the data, but you leave some data out. And then you say, how well is my classifier? You can evaluate that by looking at how they do on the left out samples. And that gives you a rough idea how much your model or your classifier, how much errors or false positive or false negative this classifier has. Then you can, of course, use all the data to train, but then you will know that when you test on new samples, you probably have this level of error rate, okay? And then we started doing next generation sequencing. With next generation sequencing, the raw data you are going to get is the FASTQ file. And you have already seen that in homework 2. For quality control, we do FASTQ-C just to make sure, you know, the read towards the end, the quality don't drop too quickly, depending on how long you sequence. And then we discussed read alignment. Right, so the most commonly used read alignment algorithm is BWA and also bowtie. Both of them are widely used, and both of them use the Boros-Weller transformation. Sorry, there's a typo. It should be BWT. And then in the alignment, we are using this LF mapping in order to really, you know, from the top to the bottom, gradually figure out where the hits are, the mapping locations are. And after the alignment, you will get a SAM file, which can be compressed into a BAM file. And also, BAT file is a very kind of simple, just tab delimited text file of the locations. And there are some tools called like SAM tools or BAT tools, which allow you to do very quick manipulation of the SAM result or the BAT results, like overlap, union, or whatever, okay? Then we talk about Rna Seek. So Rna Seek in alignment, we need to make sure that the splice, we need an aligner that's splice aware, meaning that it knows that these are exons and there would be reads that cross between one exon to the next exon with a gap in the middle, right? By creating those junctions in the index. So we ask you to try this star in the homework. There is a competing algorithm called HiSat, which is a developed by the same group that developed the bowtie. And then in homework 3, which we should give out today, maybe it's already online, we also discuss pseudo aligners. So these don't really align to the whole human genome. It just uses the known annotated RNA transcript. So it's really fast. You only align it to the 2% of the genome. And there are also competing algorithms, Callisto and Salmon. We're going to ask you to try Salmon for homework 3. And then there are some de novo algorithm which can really give you the novel junctions, or even if you don't have the reference genome that you can do. Oh, by the way, I forgot to mention something. You might also want to do a quality control of your RNA samples, right? You want to see are the reads really landing in exons, not in the middle of nowhere, right? Are they giving you the most of the known junctions and things like that? And then in order to do quantification, there are a number of terms people use. Early days, people use this RPKM. When they started doing paradigm sequencing, they do FPKM. But really for better expression index estimate, currently the standard is you use TPM to really represent the expression of a gene. However, when you try to calculate a differential gene expression, so quantification is just how much this gene is expressed, but the long genes do have more reads. And when you calculate the differential expression, the longer genes will have better power of detecting differential expression level. And so for differential, for quantification, I think we should use TPM, but for differential expression, we should use the read count, the actual read count. You want to normalize a little bit and adjust for the like a GC content a little bit, which pseudo-aligners like Simon already do. I want to mention, if you are using star, so star alignment is still considered the most accurate. It's just a little slower. If you have the time and you really want to get some like very, very accurate result, you should use the star. So star doesn't automatically give you like a TPM. Star will just give you a BAM file. You will still need some tools like HT Seek count, from the star data to convert into the read count on every gene before you can run DE Seek downstream. But if you run Salmon, it will already be in a format that can directly run DE Seek. Okay, so in homework 3, we didn't let you try star followed by DE Seek, but just you're aware, if you do run star, you want to use this, some other way to get from the BAM file into a read count on every gene before you run DE Seek. Okay, and then for differential expression of the RNA seq, we learned that RNA seq read coverage on the genes follow a negative binomial distribution, and algorithm like DE Seek too. And I want to also quickly mention there's Lima Voom. So this is the group that developed the Lima. If you have an experiment with very complicated designs, like you knock down genes with different siRNA, and you do different replicate, like a really complicated factorial type of design, Lima Voom is probably better. But if you just have simple, most of the time, 2 conditions, you want to do differential expression with very few replicates, DE Seek is much more sensitive. And so DE Seek, they use a negative binomial distribution to estimate the kind of the parameters, and also it try to borrow information from all the other genes as well to get you a more robust estimate of the variance. Because most of the time when you do RNA seq with only 2 replicates, your variance estimation is no way going to be accurate. So it knows basically roughly when the expression is this much, the variance should be this much. When the expression is higher, the variance would be higher. So it really borrows information from all the nearby genes. In addition, in order to reduce your final multiple hypothesis, in order to test algorithm like DE Seek or EdgeR, they try to eliminate those genes that are hopeless. No way they can be differentially expressed because either they have really low average TPM, or if you have a lot of zeros, or then they are hopeless. So this way they reduce the number of multiple hypotheses to be tested, so you can get more differentially expressed genes called at the same FDR level. After you call the differentially expressed genes, you can still represent the expression of every gene using the TPM. Then you can do clustering of different genes in multiple samples using the TPM level. You just eliminate those genes that are not differentially expressed and focus on the ones that are differential. Look at how they cluster in different conditions. You can also annotate those genes as in microarrays. You put the 200 genes in David and see what you get. I want to mention, most of the time, you will get a good result if you do annotation with, I would say, over 200 genes. If you only get 50 differentially expressed genes, it's very rare that from David, you will really get some very significant pathways. And so if you don't have enough genes to run, like if you only get 36 differentially expressed gene, what you could do is you just lower your cutoff a little bit, because say FDR, previously you can say I'm using fold change of 1.2 and FDR of 0.05. Maybe you say, sorry, fold change is the log 1.2, or you say two-fold change or something, right? You can lower your cutoff a little bit. Just say maybe my FDR can be 10%. I mean, you can suffer 10% false positives maybe, right? Or the fold change, instead of 2-fold, you do 1.5-fold, just so that you have a few hundred genes to run the David. or you can then run gene set enrichment analysis. Okay. Now, next is single cell RNA seek. Yeah, so we discussed that there are two different technologies. There's Smart Seek and also droplet based. For Smart Seek is when you have very little cell, like a 1 cell embryo to 2 cell embryo, or whatever, 20 cell embryo, like really you have to put them into single cell. I think that's something you use. But if you really have a piece of tissue, like a tumor or some, like liver or blood, your droplet-based approach is much easier to process, because right now SmartSeq, you still have to buy the kit and really do it 1 cell at a time. It is more expensive. It does give you better readout. And then if you use droplet-based method, there are like 10x genomics machine, and they have this cell ranger algorithm. Remember, for droplet-based, there is a cell barcode. Based on the barcode, you know which cell is coming from, but there is also a UMI, which is used in case you have too much PCR amplification on the same initial RNA. And so with Cell Ranger, you will get this matrix, which is the read count already mapped, read count per gene per cell. It doesn't try to map the transcript, because droplet-based approach currently still have the 3' bias. You're only getting the 3', and anyways, very often you don't know the exact full-length transcript. And there, you will merge all the UMI for the same gene, for the same cell. You merge the different UMI as just one read count, okay? And so by this time, people believe that this, the single-cell RNA-seq reads, mostly they follow this zero inflated negative binomial distribution, which is, if this gene is a dropout, you have nothing. Even if the gene might be expressed at some high level, for some reason you just didn't get it at all. And so those will all just get zeros. It's only when it is not zero, then it follows kind of the negative binomial distribution. And in order to really do the differential expression test, we're gonna use a Surratt, introduce this in the homework. And SCDE is developed by Peter Kachenko's group at Harvard. And their original algorithm is called SCDE. I think now it's called Pagoda. And the second one has more point and click kind of user interface, which is very friendly to experimental biologists. And Surratt is more script-based. So first of all, you want to QC the cells. If the cell has too few transcript or too many transcript, you don't want that. Because when you have too many transcripts, it could be because of multiple cells in the same droplet, right? And also you want to QC the genes. If the gene is only showing up in 3 cells or very few cells, maybe you don't need to worry about those. And because the data is too big, we want to do a PCA to shrink the dimensionality. So each cell, instead of being represented by 5,000 genes, mostly 0, or actually, all the genes, and then many zeros, you want to shrink them to, say, 100 dimension. Because, most of the time, you want to visualize, you want to say, okay, what is the, say in the blood, what are the major cell types? Oh, there's, CD4 T cells, CD8, like basically with 100 dimension, you should be able to separate most of these. And so you can do a PCA to get, say, the top 100 or even 10 dimensions. And then you want to use the clustering. And we mentioned, we look at this before. You just look at the pairwise distance and you want to try to find the tight clustering. And then we can visualize them on t-SNE, which again, the within cluster distance, you can see the separation, but the size of the cluster sometimes is not very good. If it's really tight, all the dots really overlay each other. The distance among the far away clusters, they are not representative. Okay, so it's not like if you see these 2 clusters versus these 2 clusters, these 2 are closer. That doesn't happen. But you at least can see distinct clusters better than what you will see from PCA. And then we can look at the differential expression. So if you want to look at the differential expression between the clusters, what you can do is with droplet-based, you just average the 500 cells together in that cluster. That will give you a level pretty much okay to be compared with another situation. But Surratt has both this should be DE-Seek2 and the MAST. You can use either of the two to try this. And then Surratt also used CCA, this canonical correlation analysis, for batch effect removal. I think that's all the tools we developed. I mean, we discussed in this course. And so one thing I always get asked, well, especially for the statistics students in this course, I think this is undergraduate, if their concentration is on computational biology, they are asked to take this course. And some statistics students get really frustrated. Like, we don't do any equations, it's like very different from most of your statistic course. But if we look at just a few slides back, let's look at this situation. We can see which of the tools are developed by statisticians or biostatisticians, and which of the tools are developed by computer scientists. It's very interesting. As I teach this course, you can see RMA was developed by a statistician because it's a number, right? You have the numbers, you try to normalize, you try to estimate some, weighted average of some sort, right? And Lima is, again, you have a bunch of numbers, you're trying to evaluate the differential expression with some, batch or whatever. Yeah, so these are kind of statistical approach. Hierarchical clustering and k-means, this more, I would say, computer science like. T-SNE, PCA. PCA is more math, I would say. T-SNE is like more computer science. Initially, it was for handwriting recognition. If you have, a lot of people write many ones, many twos, many threes, and then they try to figure out whether they can recognize what it is. And they see, T-SNE can separate them well. Batch effect removal, COMBAT and SVA are developed by biostatisticians. By the way, FDR is, false discovery rate. These are all statistical approaches. Gene ontology is actually developed by biologists. They decided, okay, let's get together to annotate these samples. But really using the tools to look at the differential expression, so to annotate which gene ontology category or pathways are enriched, they are using statistical approach, right? The KS test or some, these chi-square tests, these are all statistical approaches. And then once you go to this machine learning, K-nearest neighbor is a computer science kind of development. MTS, actually, I don't even know. PCA, more math. T-SNE is more CS. And when you go to the supervised, logistic regression is a statistical approach. It's a statistical machine learning. Random forest, this tree-based, is a computer science machine learning. Support vector machine is more computer science machine learning. So you can see there's like differences. And then initially when high throughput sequencing was starting, CS people really jump on because there's a lot of data. You have to do the read mapping, alignment, find the location, look at the differences. These are all CS. FastQC is actually pretty simple. You just kind of display, but BWA, Bowtie, BAM2, these are all CS algorithms. But then once you go, after you finish, I would say STAR is more CS as well. But once you align the sequence to a gene, now you have expression index, you try to calculate difference. The group, I forgot to mention, from read mapping, an algorithm called RSAM can give you the kind of the TPM. They were the group that initially, RSAM was the group that initially decided to use TPM to get the normalized gene expression data. And the group that proposed TPM is a biostatistician. And for DE-Seek2 and Lima Wung, these are all biostatisticians. Okay, and cluster annotation, these are the same as before. And for cell ranger to UMI, this is just coding, it's like scripting. But Surat, this is the tool was developed by Rauf Sajita's group, and he's kind of interesting. He has a PhD in statistics, and now he's also doing experimental work, doing a lot of single cell experiments. But I would call this as kind of a statistical tool. SCDE is, I would say, also using more statistics to calculate these differences in here. And yeah, so for things like MAST or CCA, they are more like statistical approaches. So you can see statisticians and biostatisticians are really playing an important role in developing the methods, not for alignment, but once you have the counts, anytime you deal with counts and how to look at the differences, normalize, that's where statisticians play an important role. Okay? So that's the second part.